I welcome you all in this Science Dialogue series. As you all know, this is a free webinar series, and main purpose is to increase knowledge pool. But yes, you will also get e-certificate if you will fill e-feedback form. This Google form we will share after this lecture in chat box only. In this chat box, you can interact the speaker as maximum as you can, but generally we allow after lecture. But in meantime also you can write in chat box, or you can unmute and speak. So I request Dr. Jyoti to introduce our speaker and formally start this session.
Okay. So good evening, everyone. Myself, Dr. Jyoti Vihas Thaikkottathil. I am working as an associate professor in Institute of Advanced Research, Gandhinagar. So it is my privilege to introduce today's speaker. First, definitely I want to thank Dr. Gopal ji to start this platform where every scientist, they can share their experience about the research, and a student can benefit by listening all those experts in the various area. So today's speaker, we have Dr. A. Kalidoss from SRM Art and Science College, Chengalpattu, Tamil Nadu. He is a prominent microbiologist and biotechnologist who has made significant contribution in microbiology and biotechnology. Dr. Kalidoss has several publications, 15 research articles, 7 book chapters, and his research work has been widely cited by the other scientists as well. Dr. Kalidoss has conferred also Ratan Lal Innovative Science Award on May 2025. He is also serving as advisory board. He has in peer reviewer. He is a peer reviewer and in editorial board member in several national and international journals. Dr. Kalidoss has presented his work, his research work in several national and international conferences, and also won Distinguished Speaker Award as well as Best Presenter Award in those conferences. Dr. Kalidoss is an educator. He has demonstrated deep commitment for his mentorship and student development and fostering the collaboration and encouraging the dynamic academic environment. He has supported various student and faculty, inspired various student and faculty, and contributed overall their advancement in microbiology and biotechnology. Dr. Kalidoss has also received Best Inspiring Teacher Award recently in September 2025. He has also served in various organizing committee as a resource person, as a chairperson, and as a keynote speaker in various conferences, workshops, and seminars. So without delay, we'll request Dr. Kalidoss to present his work and share his knowledge with the reviewers here, attendees, or most of the students probably. So we welcome you, Dr. Kalidoss. You can present your lecture now.
Yeah, thank you.
Thank you.
You can share the slides.
Yeah, sharing. Is the slide visible?
Yes.
Yeah.
Yes, it's visible.
Okay, sir.
Yes.
Is the slide changing now from one slide to another? Is the slide changing from one
Yes,
slide to
yes,
another,
yes.
sir?
Changing.
Yeah, okay. Okay. Good evening, everyone. First of all, I thank our respected principal, Dr. Vasudev Raj, sir, and respected HOD, sir, Dr. O.V. Ravikumar, for permitting me to act as a resource person in today's webinar. I welcome everyone to today's webinar, Strategies of Vaccine Making. It's great to see all the educators, students, and professionals all coming together to learn and grow in Science Dialogue series moderated by Dr. Gopalji. I thank Dr. Gopalji for giving me this wonderful opportunity. Today's contents for the seminar and webinar: introduction to vaccines, ideal vaccine properties, how vaccines work, principles of vaccine production, key steps in vaccine production, vaccine development cycle, how vaccines elicit immune response, prophylactic vaccines versus therapeutic vaccines, and the conclusion. First of all, in introduction of vaccines, this is general concept, a general definition about vaccines. Vaccine is a prophylactic treatment. Prophylactic means before the onset of the disease, the treatment is given as a preventive measure. Okay, so here a preventive treatment. A vaccine is a prophylactic treatment because its primary purpose is to prevent a disease from occurring in the first place by stimulating the immune system to build immunity. Not only the preventive treatment, preventing treatment is associated with stimulating the immune system to build immunity by the production of antibodies. Okay, so prophylaxis simply means preventive care, and vaccines work by introducing the body to the weakened or inactive form of a pathogen, so it can learn to fight it off if exposed to the real pathogen, the real thing. What are the properties of ideal vaccine? A good vaccine is safe, effective, and produces a long lasting immune response that prevents or reduces the severity of the disease. Safety and tolerability is one of the properties. Efficacy and immunity is another property. Then practical considerations and community level impact. These are the ideal vaccine properties. First, let us deal with this safety and tolerability property. Low risk of adverse effects, then minimal risk of infection. Low risk or adverse effects, here it should not cause serious side effects when it is given in the form of vaccine, and be well tolerated by a wide range of people. Okay, that is included in safety and tolerability. Then minimum risk of infection, it should not revert to its virulent form again. When it is being injected into a patient or into a human, it should not revert back to its virulence form, and it should not gain the pathogenicity again. That is another property of vaccine. Then efficacy and immunity. High efficacy, it effectively lowers the risk of disease and produces a protective immune response. Then long-lasting protection. A good vaccine should confer immunity for an extended period of time, ideally for a lifelong, requiring fewer or more spaced out doses. Then stimulates protective immune memory. It is another property that it should stimulate protective immune memory. It generates memory B cells and T lymphocytes to allow for a rapid and robust response upon re-exposure to the pathogen. Then another property is induces neutralizing antibodies. It should induce neutralizing antibodies. It should prompt the body to produce antibodies that can neutralize the infectious agent. Generates protective T cells. It should stimulate T cells that can kill infected cells or support other immune functions. And practical considerations: easy to administer. Then thermal stability, it should not get denatured due to thermal fluctuation due to heat here. Then cost effectiveness, it should be affordable to produce and distribute widely. Then multivalency, In some cases, a single vaccine might protect against multiple strain or types of pathogen. Then let us see how a vaccine works. How do vaccines work? Vaccines reduce the risk of infection by working with the body's natural defenses to safely develop immunity to diseases. A weakened or killed form of the disease is injected into the body. The body creates antibodies to fight the germs. If the actual disease germs ever attack the body, the antibodies return to destroy them. It is It is very simple. When we are injecting the vaccines, it elicits immune response in the form of production of antibodies. The body creates antibodies to fight the germs. The germs are said to be the antigens, and it is an interaction between the antigen and the antibodies here. Look at the second column in this diagram. The body creates antibodies to fight the germs. So the germs are considered to be the antigens, and it is the interaction between the antigen and antibody. If the actual disease germs ever attack the body, the antibodies return to destroy them. Okay, obviously the antibodies return to destroy the antigens here. Principles of vaccine production. The principles of vaccine productions involve identifying and cultivating the pathogens antigens. Whatever the vaccine may be, the principle is very simple. You have to check for lack of pathogenicity, and it should retain immunogenicity. So this is the key principle here. It should lack pathogenicity. The source organism which you are selecting can be either bacteria or virus. It should lose its pathogenicity. That is, disease-causing ability should be lost, and it should elicit an immune response when it is being injected into the host or the patient. Okay, so immunogenicity plus pathogenicity minus. This is the key principle of vaccine production. What are the key steps in vaccine production? Research and design, preclinical testing, clinical trials, regulatory review and approval, antigen preparation, formulation, quality control, packaging and distribution. These are the key steps in vaccine production. First, let us see the research and design. Scientists identify or engineer the necessary antigen. So you have to identify the antigen, suitable antigen. Hope you know the basic definition for antigen. Let us take the term antigen and split it into two, anti and gen. Anti refers to antibody and gen refers to generators. Antigens can otherwise be called as antibody generators. Antigens, when it is being injected into the host, it should elicit an immune response in the form of antibody generation. Hence, you take the sentence antibody generation, and you can construct the word antigen. Hence, antigens are foreign substances which has the ability to generate antibodies. Okay, so here antigens, protein, sugars, or nucleic acids are selected here by the scientists. They are identified, and they are using techniques like genetic engineering or cultivation. Then preclinical testing. Safety and effectiveness are assessed in the laboratory settings and animal models to evaluate the antigen stability and immunogenicity. So in the preclinical testing, safety and effectiveness of the vaccine is checked or assessed in the laboratory settings. And in the animal models also, it is again rechecked to evaluate the antigen's stability. The antigen which we are injecting, it should be stable. It should have the ability to induce the immune response. It should immune response in the form of antibody production. Then clinical trials. Then the next step is clinical trial. human trials are conducted in phases to further assess the safety, determine dosage, and confirm effectiveness in large populations. Then regulatory review and approval. Based on clinical and manufacturing data, regulatory agencies review the vaccine for safety, efficacy, and quality before granting approval. Then antigen preparation, the antigenic component, be it protein or sugar or nucleic acid, whatever the antigenic component may be, it is harvested and purified to remove the impurities. You have to purify the antigen at this step in antigen preparation. For example, this can involve harvesting viral particles from cell cultures or antigens from yeast cells. Then formulation. Here comes the formulation of the vaccine, the purified antigen. In the previous step, we are purifying the antigen. Here, in the next step, the purified antigen is combined with the necessary ingredients along with excipients. Okay, the purified antigen is combined with other necessary ingredients. These can include adjuvants to enhance the immune response, stabilizers are added, then preservatives are added, diluents are added, and excipients are also added. Excipients have not included here, but excipients are also included in formulation at the as a last one in the ingredients. Then quality control. Throughout the manufacturing process, meticulous quality control measures should ensure the vaccine is safe, pure, and potent. Then packaging and distribution. The final product is filled into sterile vials or syringes and packaged for storage and distribution with cold chain management crucial to maintain potency of the vaccine. So these are the key steps in vaccine production. Then let us have a look at the vaccine development cycle. Okay, now look at the cycle here, and in the 12 o'clock position is numbered as 1. Selecting the strains for vaccine production is the first step, 12 o'clock position. Then step number 2 is cultivation of microorganisms. Then isolation and purification of microorganisms is the next step in the development cycle. Then inactivation of the isolated microorganisms, then formulation of vaccine, then quality control and lot release. So uh, these are the vaccine steps which are involved, key steps which are involved in vaccine development cycle. Let us see in detail about selecting the strains. Manufacturing begins with small amounts of virus or bacteria, which is actually said to be seed. Okay. Virus and bacteria used in the manufacturing can be selected from a seed lot system. The record of origin, passage history, storage conditions should be maintained for each seed lot. The virus must be free from impurities, including other similar viruses. The seed must be kept under ideal conditions, frozen, that prevent the viruses from becoming stronger or weaker than desired. Here, this is very vital to keep the seeds under ideal storage conditions. Even if the virus is getting stronger, or even if it is getting weaker than desired, then it becomes either virulent or much stronger. It will give us undesired results when we are injecting it into the host cells or into the patients. So it is very crucial to keep the seeds, that is either virus or bacterial seeds under ideal conditions. It should be frozen and kept for storage. Stored in small glass or plastic containers, the choice of the seed depends on a number of factors, including the efficacy of vaccine and its secondary effects. The bacterial strain or cell line should be obtained from a recognized culture collection, one with an established and documented provenance. If the chosen virus is in-house clinical isolate, it is necessary to compile a complete history of the strain. That is, you have to check the origin, passage history, everything. Okay, everything about. If it is an isolate, you should know the complete history of the strain, right from the origin. From where did you isolate? Uh, from which kind of patient you have isolated it. So you know the history of the strain. Okay, so these are all the things which are involved in selecting the strains. Manufacturing begins with selecting small amount of virus, bacteria, or any microorganism in this case. Then cultivation of microorganisms. In the previous step, we have selected the microorganism. It can be either virus or bacteria. Okay, so here, if it is a bacteria, how will you cultivate? If it is virus, how will you cultivate it in the next step? So as we all know, as you are aware of the thing, viruses cannot be cultured just like bacteria with the help of media. With the help of media, we cannot cultivate viruses. So it needs specialized techniques like cell cultures. You can cultivate the viruses in birds embryo, then live animal inoculation. Then you can cultivate the viruses by using transgenic animals. You can create transgenic animals, and you can incorporate the viral gene into the transgenic animals, and you can incorporate the vaccine property also transgenic animals. And cultivation of bacteria methods used are batch culture, continuous culture. In batch culture, the microbe is grown in a closed vessel, then typically either in a test tube or in a conical flask. Then continuous culture, it is performed with chemostat. The microbe is grown in a vessel which has medium constantly added and spent medium constantly removed. Okay, in industrial microbiology, you should have studied this chemostat in detail. Okay, continuous culture. Okay, it is in this continuous culture, the freshly prepared medium is added on one side, and the spent medium is removed on the other side. It is performed in a chemostat, and by using this, you can cultivate the bacteria. Then isolation and the purification of microorganisms. So look at the previous two steps. You have selected the strains, you have cultivated the microorganisms, and the next step is product isolation and purification here. Product isolation is the removal of those components whose properties vary markedly from that of desired product. Say, for example, from bacteria, if you are isolating only a particular component, that component is mentioned here in product isolation here. Okay, if you want to use any one of the cellular components as antigen, that is considered to be a product here in product isolation, is the removal of those components whose properties vary markedly from that of other desired product. Then purification. Purification selectively separates and retains the desired product at the highest purity per its predetermined specification. It is also involved in removal of unwanted compounds. Okay, purification, it is just focusing on desired compounds. If you want only spike proteins from a virus, then that is our main focus, and other things are considered to be other impurities. Other things are considered to be impurities here. Removal of unwanted compounds. You have to remove those unwanted compounds. You have to retain only the spike protein in this case. The most common method of vaccine production is based on an initial fermentation process followed by purification. Then for purification, we can adapt either centrifugation, filtration, chromatography. All these three techniques are used here. for the purification. Let us see centrifugation. The principle behind centrifugation is sedimentation. Everyone knows, I suppose. Centrifugation is a process by which solid particles are sedimented and separated from a liquid using centrifugal force as a driving force. Centrifugation is used to separation and purification of pathogenic virus antigens and other agents used in the production of vaccine. So the main purpose is to remove the impurities based on the density you are segregating. You are purifying. You are purifying here based on the weight. The impurities are getting separated out. Only the desired antigen is retained here in centrifugation. Centrifugation is also used to remove dead cells, cell debris. Example: influenza vaccine, rabies vaccine, hepatitis B vaccine, and Japanese encephalitis vaccine production. In this vaccine production technique, centrifugation, by using this centrifugation technique, it is purified here, purified. Differential centrifugation can also be used. Density gradient centrifugation can also be used. Rate zonal centrifugation, isopycnic centrifugation are the other types of centrifugation. All these can also be used for the purpose of purification in vaccine making. And when we talk about chromatography, a group of physical separation techniques which are characterized by separation of mixtures due to differences in the distribution of coefficient of the sample components. This is the basis of chromatography and principle of chromatography, that is separation of mixtures due to differences in the distribution coefficient. So this is the key principle of this chromatography. Coefficient, distribution coefficient. Based on the distribution coefficient, we can separate the components, several components. Between two phases, one phase is stationary and another phase is said to be mobile phase. Example: modified vaccinia and coronavirus and smallpox vaccine. In this vaccine making, chromatography is used for purification. For the purpose of chromatography, we can choose another type of chromatography like ion exchange chromatography, affinity chromatography. All these can be used for the purpose of purification of antigens. Purification means here our focus is to um, our focus is only on the antigen. The antigen can either be the complete organism or it can be a cellular component itself. If in the case of cellular component, your focus is on the cellular component and other things are considered, the rest of the cellular components are considered to be debris and we have to remove that. Our target is only on the cellular component only. So that is our antigen which we are going to use for vaccine making. That is considered to be antigen. So by means of separation, we are selecting the antigen here, purifying the antigen. Then filtration is another purification method. Separation of particles from liquid by applying a pressure to the solution to force the solution through a filter. So you are passing the solution through a filter. Filtration is classified in two ways: dead end filtration, tangential filtration. So dead end filtration, let us see this one in detail now. All the flows are directed through the membrane with material building up on the surface or filter. As these particles build up, flow through the filter is quickly reduced and finally it ceases completely, causes buildup of filter cake on membrane. Okay, so here by using this filtration, we are separating only the desired antigen and other things are retained within the filter itself. Then tangential flow filtration, cross flow technology. During this cross flow filtration technology, cultured fluid is recirculated in tangential flow parallel to the filter membrane. Built Buildup of viral particles on the membrane is minimized by the recirculation of fluid over the surface, which also facilitates the concentration of particles present in the retained fluid, mainly used in purifying inactivated above-viral antigen. Then ultrafiltration, a technique for separating dissolved molecules in solution on the basis of size, rating the particles will be retained at the surface of the membrane. During this process, desired proteins and their allied products are separated with their molecular weight and volume is reduced, thereby increasing the purity of the antigen, which we are going to use it for the purpose of vaccine making. So this is the purification in vaccine production. You can say it as a purification train or a purification scheme. Fermenter from then harvesting is the next step. It enters into the dead end filter, then tangential filter, then ultrafiltration is done. Chromatography columns. It passes through the chromatography columns and it reaches the ultrafiltration. Then size exclusion column. It passes through the size exclusion column. In this way, we are purifying and thus getting the desired product, which we are going to use it for the vaccine production here. Viral inactivation is done. Then ultrafiltration, again, it is passed into the ultrafilter 3. Then formulation is done. Then excipients. Then the desired vaccine is recovered. Then inactivation of microorganisms. So what do you mean by inactivation here? Inactivation refers to attenuation here. Attenuation. Okay. We all know that the very first step in vaccine making is isolation of the causative organism or isolating the pathogen. Then we have to inactivate the microorganism. So it is crucial to inactivate or attenuate the causative organism here. Let us see it in detail. Killed or inactivated vaccine, virus inactivation. Viruses can be lipid-coated or non-enveloped. Virus inactivation involves dismantling a virus's ability to infect cells without actually eliminating the virus. Then virus inactivation works by one of the following two mechanisms: by attacking viral envelope or capsid and destroying its ability to infect or interact with cells, by disrupting the viral DNA or RNA and preventing replication. If you are targeting the replication, then it cannot multiply further. Okay, the DNA is prevented from replication. The RNA is prevented from replication here. Thus, the virus is being targeted and thus it is inactivated here. By attacking the viral envelope, you can target the virus. You can target the virus and you can inactivate it. You can also target the capsid and also you can destroy its ability to infect and interact with the cells. If you have weakened the virus, then it loses its pathogenicity and only the immunogenicity is retained here. So this is the key principle here. It follows the key principle which have previously discussed in this slide. In this particular slide, we have discussed about the principles of vaccine making, pathogenicity and immunogenicity. It should lose its pathogenicity and it should retain its immunogenicity. So this is the key principle, and this principle is followed in all the vaccine making. And here in killed or inactivated vaccine making, we are following the same principle. So in these two steps, we are weakening or attenuating the virus in order to lose the pathogenicity, and we have to check for the immunogenicity, whether it is producing whether it is eliciting an immune response, and whether it is supporting the production of antibodies in the host or not. They have to check that by in the next steps we have to check that. So this inactivation can be done either through solvent, detergent inactivation, pasteurization, acid pH inactivation, low pH treatment, then ultraviolet treatment, radiation treatment also. Let us see in detail about solvent detergent inactivation, SD inactivation. This type of inactivation is effective with lipid-coated viruses. The detergents used in this method disrupts the interactions between the molecules in the lipid coat, rendering the coat dysfunctional and impeding replication. So here, when we disrupt the interactions between the molecules in the lipid coat, the coat dysfunctional, and it obviously affects the replication here. Most enveloped viruses cannot live without their lipid coating. We all know that viruses are acellular organisms, and it is enveloped with this kind of lipid coat only. And if we are targeting this lipid coat, these enveloped viruses cannot live. So they die when exposed to these detergents. So when we use these detergents, the lipid coat breaks down, and further the virus cannot survive. And the components within this the virus particles can act as vaccines then. So this is one of the key strategies using this SND inactivation. Other viruses may still live, but they are unable to reproduce, rendering them non-effective. The detergent typically used is Triton X100. Triton X100 is an example for detergent, and this is used for the purpose of SD inactivation, solvent detergent inactivation. Then pasteurization is another technique which is used for inactivating the microorganism, effective for both non-lipid and lipid-coated viruses, because pasteurization involves increasing the temperature of solution to a value that still sufficiently denature the virus. It doesn't matter whether the virus has an envelope or not, because the envelope alone cannot protect the virus from such high temperature at 60 degrees Celsius for 10 hours. So pasteurization is another effective technique for inactivating the microorganisms for the purpose of vaccine making. So because pasteurization involves increasing the temperature of solution to a value that will sufficiently denature the virus. It doesn't matter whether the virus has envelope or not, because the envelope alone cannot protect the virus from such high temperatures. Then inactivation of microorganisms by using acid pH, low pH treatment. It is otherwise said to be low pH treatment, most effective with lipid-coated viruses. Acidic conditions deactivate the viruses. The viruses require, like other microorganisms, do require a suitable or optimum pH for its cultivation. And here in this strategy, we are modifying the pH here, bringing down the pH to acid. Okay, low pH treatment. Acidic conditions deactivates the virus. Okay, when we are using the acidic conditions, the virus cannot survive, and thus we are deactivating the viruses. Then incubation typically occurs at a pH of 4 and lasts anywhere between 6 hours and 21 days. So this is how we should use this low pH treatment for inactivating the microorganisms. This technique can also be used not only for viruses, this technique can also be used for bacteria also. Primarily, we are discussing here with the virus concept, but this is applicable for bacteria also. Then radiation inactivation, UV inactivation. UV rays can also be used to inactivate viruses since virus particles are small and the UV rays can reach genetic material, inducing the dimensional of nucleic acids. Once the DNA dimersed, the viral particles cannot replicate their genetic material. So here, when we are exposing, thymine dimer is formed here. That is said to be DNA dimersed. The virus particles cannot replicate their genetic material. Due to thymine dimer formation, the virus particles cannot replicate further. Nucleic acid. Nucleic acids, here extraction of nucleic acid. Nucleic acid is obtained from collected and lysed cells. The nucleic acid is purified by solvent extraction and chromatographic techniques and formulated for the final vaccine product. Nucleic acid vaccines can be regions of RNA, either regions of RNA or DNA that code for disease-associated proteins. Then inclusion bodies can also be used for vaccine making. Bacterial cells often are used to produce proteins that can function as vaccines. Bacteria produce proteins intracellularly, either intracellular, and store the produced proteins in the internal structures called as inclusion bodies. Following bacterial cell collection and lysis, the inclusion bodies are collected and disrupted. This often involves a series of steps involving protein denaturation, followed by protein renaturation or folding. Filtration is employed to achieve clarification of the protein solution during the process. After the extraction of these inclusion bodies and the proteins, filtration is employed here for the purification, of the protein, which is said to be clarification here. Clarification implies purification of the protein solution. Then membrane extraction. Vaccine products can be proportions of bacteria or mammalian cell membrane structures. These membrane structures are typically protein but can be lipid or carbohydrate molecules. These membrane components are usually associated with the disease state. The vaccine product is formulated from the extracted and purified membrane structure. Capsule extraction. Some bacteria grow and secrete a complex carbohydrate material, forming an external capsule. This capsular material can also be isolated and purified to formulate a vaccine. The capsule extraction process usually requires multiple steps of solvent extraction, followed by chromatographic separation or other standard purification techniques. Then let us have a look at the live whole vaccines. Several methods have been used to attenuate viruses for vaccine production. Used use of a related microorganism from another animal. Then use of a related microorganism from another animal is one of the strategies used here for vaccine making. Then administration of pathogenic or partially attenuated microorganism by an unnatural route. Okay, let us suppose if a microorganism is infecting through respiratory tract, through inhalation, okay, and you are administering it through ingestion, okay, then this is an example here for the example B, administration of pathogenic or partially attenuated microorganism by an unnatural route. Then passage of microorganism in an unnatural host or host cell. Yes, this can be explained in this way. That is, each and every virus or each and every pathogenic microorganism has some specificity. For example, rhinoviruses is very specific to human beings only. It is very specific. Rhinovirus can cause common cold only in human beings and not in other animals. So here, cultivating such microorganisms in unnatural host increases the immunogenicity also. This is another strategy which is used here in vaccine making. Then development of temperature sensitive mutants. Okay, temperature sensitive mutants is another strategy here in live whole vaccine making. Let us see in detail about this use of related virus from another animal. The earliest example was the use of cowpox to prevent smallpox. So this is a well-known example here. There is a very earliest example, use of cowpox to prevent the smallpox. Okay, to prevent the smallpox, we all know the history here. Okay, history, and for the prevention of this smallpox, cowpox virus were used here. Then administration of pathogenic or partially attenuated virus by an unnatural route. The virulence of the virus is often reduced when administered by an unnatural route. This principle is used in immunization of military recruits against adult respiratory distress syndrome using enterically coated live adenovirus type 4, 7, and 21. Live whole vaccines, passage of the virus in an unnatural host or host cell. The major vaccine used in man and animals have been derived this way. After repeated passages, the virus is administered to the natural host. The initial passages are made in healthy animals or in primary cell cultures. There are several examples for this approach. The 17D strain of yellow fever was developed by passage in mice and then in chick embryos. One more example, polioviruses were passaged in monkey kidney cells and measles in chick embryo fibroblasts. Look at these examples. You can understand the concept of passage of virus in an unnatural host. Okay, you are changing the host here. Instead of injecting the causative organism in the original host, you are changing the host here. This is unnatural, that is unnatural host. Look at these examples. Poliovirus were passaged in monkey kidney cells and measles in chick embryo fibroblasts. Then human diploid cells are now widely used, such as WI38 and MRC5s. Development of temperature sensitive mutants. Some strains are attenuated by sudden change in temperature. There are, yes, we all know that microorganisms require optimum temperature for its growth. And here, that is used for, that factor is used for developing the temperature sensitive mutants here. Some strains are attenuated by sudden change in temperature. If you are changing the temperature suddenly, then it leads to mutation, it leads to genetic changes in the microorganisms. Okay, microorganisms. This method may be used in conjugation with the above method. Formulation of vaccines. Vaccine formulation is the process of combining the active ingredient with other components like adjuvants, stabilizers, preservatives, diluents to create a safe and effective product. Here, suspending fluid is not mentioned. Suspending fluid is also used here. This process ensures the vaccine is stable for storage, effectively stimulate the immune system, and is safe for administration. Key steps include selecting the antigen, adding necessary excipients, and rigorous quality control throughout the production. So here, vaccine formulation, okay, it involves combining active ingredient. Active ingredients, so far we were dealing with active ingredients only. The previous slides, up to the previous slide, we were dealing with the antigens purification only. And this purified antigen is formulated, which means mixed with other components such as adjuvants, stabilizers, suspending fluids, preservatives, antibiotics. In several cases, antibiotics are also added. Antibiotics, why it is added? This is a vaccine. Why antibiotics are added? You can ask me this question. Yes, antibiotics are added to vaccine making in order to kill the bacterial contamination. Low doses of antibiotics are added as one of the ingredients to stop the growth of the bacteria. It is added. Then, in several cases, thimerosal, mercury-containing preservative, is also added in the vials of the vaccine. Okay, here let us see the key components of vaccine formulation. Antigen, as we all know, we were discussing it previously, the purified part of a microorganism. It can be the complete organism, complete microorganism. It can be either bacteria, virus, or the poisonous substance which is produced by the microorganism, causative microorganism, which can be considered as antigen here. It should be purified, and this purified antigen is mixed with other components like adjuvants, stabilizers, preservatives, suspended fluid, then antibiotics, thimerosal, excipients, all these things here. Then what are adjuvants? Substances that help boost the immune response to the antigen, making the vaccine most effective. Adjuvants are the components or the substances that help boost the immune response to the antigen. Okay, it acts along with the antigen. It helps in boosting the immune response, making the vaccine more effective. Then stabilizers are added to prevent the vaccine from degrading. Okay, from degrading during the storage and to maintain its potency. Stabilizers are used for the purpose of increasing the viability here. It helps in preventing the vaccine from degrading. Okay, degrading. So it is just acting like one more preservative, stabilizing. Okay, throughout the process of vaccine making, it should be stable. Okay, it should not. Mm, it should. It should retain its property, and it should be very stable. Okay. Then preservatives. Preservatives, common examples are albumin, phenol, glycine are examples. In several cases, monosodium glutamate, okay, MSG, is also added as preservatives. So what is the purpose of adding preservatives? Prevents the growth of the bacteria or other microorganisms that could contaminate the vaccine, especially in multi-dose vials. So in order to prevent the growth of the bacteria, few toxic substances like mercury containing preservatives such as thimerosal is also added here. The idea is to minimize the contamination in the vaccine. Okay, diluents, a liquid like sterile water used to dilute the vaccine to the correct concentration before administration. Formulation process, antigen production, purification, and quality control, combining the components, and filling and packaging. Antigen production, it involves the process begins with creating or isolating the antigen. This can involve growing and then weakening or inactivating a pathogen or using genetic material to code for a specific antigen. Then purification and quality control. The antigen is purified to remove impurities. The entire formulation is then subjected to strict quality control measures to ensure its safety, potency, and purity. Then combining the components, the purified antigen is combined with other necessary ingredients like adjuvants, stabilizers, and preservatives. This we have seen in the previous slide. Okay, there is formulation, key components in vaccine formulation. We have seen that, and here the filling and packaging. The final formulation is filled into vials or syringes under sterile conditions. Packaging is designed to maintain the vaccine's stability and potency during storage and distribution, often requiring a cold chain. Then quality control of vaccines. Quality control of vaccines involves a rigorous set of tests at every stage of production to ensure they are safe, effective, and consistent using methods like in vivo and in vitro assays, mass spectroscopy, chromatography to check for purity, potency, and the absence of impurities. This process is overseen by manufacturers and independent laboratories. Key aspects include in-process testing, finished product testing, and maintaining a stable cold chain throughout the distribution. So what are the key aspects of this vaccine quality? Testing and assurance, safety and efficacy, in-process and finished product testing, finished product. Let us see the testing and assurance. Quality control is a continuous process throughout, throughout manufacturing, right from the initial development to the final lot. Then safety and efficacy. The primary goal is to guarantee that the vaccine is both safe for the recipients and effective at providing the intended immune response. In-process and finished product testing. In-process tests are conducted during production to monitor the process parameters, check for impurities, and ensure the quality of materials used. The finished products tests confirm that the final vaccine batch meets all the specifications for safety, identity, potency, and purity before it can be released. Then types of quality tests. In the quality assurance, these tests are carried. The first one, potency test, then purity test, then interference test, then identity test, then other tests are also performed. First, let us see the potency test. Measures potency test is used to measure the vaccine's ability to produce an immune response using either in vitro assays or in vivo assays. So it checks the potency of the vaccine, that is, it measures the vaccine's ability, how far it is inducing the immune response in the form of antibody production. Okay, either in vitro assay or in vivo assay. Then purity tests. After we all know that during the process of vaccine making, in the initial stages, in the upstream process itself, it is checked for its purity. It is purified. We know that. And finally, once it is getting converted into a product that is vaccine, it is subjected to purity test again. Okay, here also. Ensure the vaccine is free from contamination, includes residual toxic compounds or unwanted viruses or bacteria. Okay, only traces or residual toxic compounds are allowed here, and that is checked in the purity tests. Then interference tests for products with two or more antigenic component tests must be confirmed that there is no interference between individual components. Then identity. This interference test, I will continue with this interference test here. Let us suppose a vaccine consists of two or more products, you have to check and confirm in this interference test that the two or more compounds, ingredients, are not cross reacting with each other. Okay, if they are cross reacting with each other, then again that is harmful. The vaccine will turn to a harmful, and it will affect the host. It will affect the host, and that is the purpose of interference test for products, for vaccines with two or more antigenic components. This must confirm that there is no interference between the individual components or ingredients. Then identity tests verify that the correct active components are present. Then stability test. Stability studies are required to establish the validity of the experiment that appears on the product package. Then other tests, including checking for appearance, pH, and the absence of bacterial contamination, are tests in other tests, or tested in other tests. Then lot release. This is the final stage here. Once the product undergoes quality tests. Here, the lot is ready to release, batch or serial release for distribution. Prior to release, the manufacturer must test each batch and serial for purity, safety, and potency. Batch or serial purity test. Purity is determined by testing for a variety of contaminants. Tests to detect contaminants are performed on master seeds, primary cells, master cell stocks, ingredients of animal origin if not subjected to sterilization, and each batch of final product prior release. Then a batch or serial safety test. Batches are considered satisfactory if local and systemic reactions of vaccination with batch to the released are in line with those described in the registration dossier and product literature. Then batch or serial potency test. This is required for each batch prior to release, are designed to correlate with the host animal vaccination challenge efficacy studies. Then other tests at the time of lot release, depending on the form of vaccine being produced, certain tests may be indicated. These tests may concern the level of moisture contained in the desiccated products, the level of residual inactivate in killed products, the complete inactivation of killed products, pH, the level of preservatives and permitted antibiotics, physical stability of adjuvant, then retention of vacuum in desiccated products, a general physical examination of the final vaccine. So these are the other tests in the lot release time. Then let us see how live attenuated vaccines are acting and how it elicits immune response here. Live attenuated vaccines. So what are live attenuated vaccines? We all know that live vaccines are wild viruses or bacteria